Good evening, students. Good evening, all of you. So, welcome to Unacademy EAMCET YouTube channel, students. So, myself, Dr.Rajendra Prasad, biology educator on Unacademy platform. So, in this class, we'll be discussing about the EAMCET 2020 question paper. So, that is the part 1. So, I'm going to discuss about the question paper, that is EAMCET question paper that was conducted on 25th September in this particular class. So, I'll be discussing nearly 20 questions. So 20 questions I am going to discuss. So let us see the question paper in this particular class. So before starting the class, I would like to introduce about our Unacademy Plus subscription students. So every day Unacademy is providing free live classes on its Unacademy platform, that is called Unacademy Special Live Classes. Apart from that, Unacademy is also providing free live classes on its YouTube channel. Every day at 4 p.m. we will be having the biology class on our Unacademy YouTube channel, students. So I suggest you to subscribe the YouTube channel so that you will be getting the timings of our classes. So you will be having mathematics, physics, chemistry and biology classes every day. So if you subscribe to the channel, so you can click on the bell icon so that you will get the details about our timings of our classes and topics that our topic students are going to discuss. So apart from the YouTube channel, so on our website, Unacademy Plus classes. So Unacademy Plus classes are also conducted. So what is the importance of this Plus courses is, so a designed course for the complete preparation of medical entrance examination will be conducted on the Unacademy Plus courses. So let us see what are the benefits of taking the Plus subscription. So the premium content. So benefits are premium content will be available at affordable price. So structured live courses, dedicated doubt clearing sessions will be conducted, and quizzes will be conducted at regular intervals, and batches. So batches. So lectures will be batches. You can choose the batch of lectures which you are interested, and test papers and test series will be provided, and daily practice papers also will be available to students. So this all you can get for single subscription. So if you use my code RAJENDRALIVE, so this all you can get for single subscription of 1 month trial period, that is of 2,250 rupees. So for 1 month trial price it is 2,250 rupees. Okay, students. So there is one batch has started in this month, that is in the October 12th. If you see here, so this batch, so this is the Telugu batch. Especially all the teachers will be teaching the classes in our native language, that is in the Telugu. So what is the importance of this particular Telugu batch is, so they will be covering the course to complete have complete preparation for EAMCET 2020. So detailed explanation in Telugu language for better understanding will be given, and complete syllabus will be covered. So if you are interested for this particular batch course, so you can go for 1 month plan, that is 2,250 rupees, so that you can go for the batch course. Apart from that, there is another batch that is also going on, that is called comprehensive batch for EAMCET. So in the comprehensive batch, I will be taking the biology classes, and Devi Professor will be taking the physics classes, and Sachina sir will be taking the chemistry classes. What is the importance of this batch is, a complete syllabus will be covered, tips and tricks will be given by educators, and topic wise practice questions also will be given. Full length mock tests also will be conducted to students. So 1 month plan. So 1 month plan is available, that is 2,250 rupees. It charges about, and if you use my code RAJENDRALIVE, you will get the course for 2,250 rupees. Apart from 1 month plans, so we are also providing 6 months plan as well as the 12 months plans. So you can go for the 6 months plan and 12 months plan. These are the long term plans. So the 6 months plan costs about 10,125 rupees, whereas 12 months plan costs about 12,375 rupees. So what I suggest is, I suggest you to take the 6 months plan and 12 months plan. So that are the best plans, because they are the long term plans. You can practice for a long time. So you can use my code RAJENDRALIVE so that you get the 6 months plan for 10,125 rupees and 12 months plan for 12,375 rupees. So that is about our Unacademy Plus for students. So let us continue with our topic in today's class. So let us discuss about the 25th September EAMCET question paper that was conducted on the 25th September. So let us see the questions. So let us discuss about the questions. Let us see the questions that was given in the 25th September question paper. So let us see the first question. So cilium and flagellum. So cilium and flagellum emerges from the centriole-like substance. He is telling the centriole-like substance, but it is not a centriole. So that is an important point you have to remember here. So it is telling that emerges from centriole-like substances. It was given that it is emerging from centriole-like substance, but it is not emerging from the centrioles. So that is the reason you have to read carefully this question. So cilia and flagellum emerges from which location? So first let us see the first option. So plasma membrane. So cilia and flagellum does not emerge from the plasma membrane. So plasma membrane cannot be our answer. Then you see the basal bodies. So no doubt about it. So cilia and flagellum arise from the basal bodies. So this is the right option. Apart from that, axoneme. So what is this axoneme? Axoneme is something different. So this is nothing but that is the internal structure. It is the internal structure of. So what is an axoneme? Is internal structure of cilia and flagella. So internal structure of cilia and flagella is called as axoneme. Actually means this axoneme. So when we cut the cilia and flagella, so we cut the cilia and flagella horizontally. Then we will observe that cilia and flagella which was cut horizontally, and we observe under microscope. Then we can see that there is a specific structure is observed under the microscope. That specific structure is called as an axoneme. So axoneme, what is an axoneme? It is an internal structure of cilia and flagella is axoneme. So the question is asking about the cilia and flagella is emerging from. So they are emerging from the specific structure that is called as the basal body. So finally the basal body will be our answer. Okay. Let us move to the next question, students. So welcome to few more students. Welcome to Abjit. So today we will be having maths class. So we will be having maths class at 7:30 I think. So you can just check the schedule on our Unacademy EAMCET YouTube channel once. Okay, so you can attend at that time. So let us see this question. So question is asking to match the following. So potato, so transgenic plants. So these are the list of the transgenic plants: potato, tomato, basmati rice, and soya bean. So these are the transgenic plants, and they are resistant to the which organism. That's what the question is about. Let us see. So potato, if you see the transgenic potato, so this transgenic potato, it is resistant to the fungus. The name of the fungus is called as a Phytophthora infestans. So Phytophthora infestans is a fungi. No doubt about it, it is a fungi. So Phytophthora infestans. So it's called late blight of potato. So the Phytophthora infestans causes the disease in potato, and the name of the disease is called as a late blight of potato. Okay, it's causing a disease. The name of the disease is called as a late blight of. So it is causing a disease. The name of the disease is called as a late blight of potato. So, but the transgenic potato. So transgenic potato is resistant to the Phytophthora. So it will not cause the disease to this particular transgenic tomato. Okay. So anyhow, so like that, so potato is resistant to Phytophthora. Similarly, tomato, if you see tomato, so tomato is resistant to the Pseudomonas. So what is Pseudomonas? Pseudomonas is a bacteria. So tomato is resistant to the bacterial infection. So it is resistant to the bacterial infection, specifically the tomato. Similarly, the basmati rice. So basmati rice is resistant to the biotic and abiotic stress. So basmati rice is resistant to the biotic and abiotic rice. And next, if you see the soya bean, so roundup soya bean. So soya bean is a herbicide tolerant. So based on that, we have to identify the answer. So no doubt about it. So based on that, we have to identify the answer. So once again, if you see, so transgenic plant. So transgenic potato is resistant. So transgenic potato is resistant to the fungi. So the name of the fungi is called as a Phytophthora infestans. If generally this Phytophthora infestans causes a disease, the name of the disease is called as the late blight of potato. Similarly, tomato. So tomato also causes. So tomato is infected by a bacteria. The name of the bacteria is called Pseudomonas. So transgenic tomato is resistant to the bacteria that is Pseudomonas. Then basmati rice. If you take basmati rice, that basmati rice also is resistant to the abiotic and biotic stress. Then similarly soybean. So soybean is a herbicide tolerant. No doubt about it. Soybean is a herbicide tolerant. So based on that, we can take the option as our answers. Let us see the options. So potato is 3. So where we can potato is 2 here. Find out the resistance. So let us see the option 2. Then B is 3. Okay. So this will be our answer actually. C will be 4, yes. D will be 1, right? So this one will be our answer. Next conditions, epipetalas. So epipetalas and syngenous stamens. So what is, before going to the answer, let us see what is epipetalas condition. So what is this epipetalas condition? So you can see what is this epipetalas condition here is. So here the stamens. So epipetalas condition is discussing about the stamens. Stamens are fused. So stamens are fused. No doubt about it. Stamens are fused. So stamens are fused with. So stamens are fused with the petals. So stamens are fused with the petals. When stamens are fused with petals, such condition is called as epipetalous. Stamens are fused with petals. Such condition is called as a epipetalous. Then there is another condition. Then another condition was given here in the same question. It is called as a syngenous. Okay. There is another condition that is called as a syngenous. So there is another condition that is called as syngenous condition. So what is this syngenous condition? That is the thing. So here, in this case also what happens, this is also discussing about the stamens in a flower. So this also describes about the stamens in a flower. So if you see epipetalous condition, for example, there is a petal like this. So for example, this is a petal, to that stamen is fused. So stamen is fused. So stamen is fused like this. Such condition is called as a epipetalous. Now such condition is called as epipetalous. What is about the syngenous condition? Here also it is also describing about the stamens only. So what happens is, so stamens will be having anther. So here like this. So anthers are fused. So what is syngenous condition is in a flower. So you can see this is a stamen. So in this, so what is the syngenous condition here is, what is syngenous condition is, so in a stamens, the anthers are fused, the head part, that is anthers are fused and filaments are free. Such condition is called as a syngenous. So both epipetalous condition and as well as syngenous condition, both are with respect to the androecium, that is about the stamens, no doubt about it. Then what is epipetalous condition is, in a stamen, that stamens in the flower, that stamens are attached to the petals of the flower. Such condition is called as epipetalous. Then what is syngenous condition? So here in a stamens, in a bunch of stamens, the anthers are fused. You can see here the anthers are fused here. So anthers are fused. Anthers are attached to one another. So anthers are fused and filaments are free. So anthers are fused and filaments are free. Such condition is called as a syngenous. So generally, in which type of members we can see the syngenous condition and epipetalous together condition is, we can see in the Asteraceae members. So Asteraceae members generally exhibits the epipetalous and syngenous stamens. Okay, so both conditions, syngenous and as well as epipetalous conditions, both are observed in case of the family members like Asteraceae. In Asteraceae members, we can see both epipetalous and syngenous stamens. So let us see the next question. So functional xylem of the dicot trees, that is the question. So functional xylem. So question is asking about the functional xylem. That is the important point which you have to remember here. So functional xylem. So let us see about the functional xylem. So what is this functional xylem actually in case of the dicot tree? So if the question is asking directly about dicot tree, we will take into consideration as a stem. So in a stem, so we have, in a stem, if you see the stem, we have the sapwood. So we have sapwood, no doubt about it. So we have the hardwood. In this term we have sapwood and as well as hardwood. So hardwood. If you see the composition of the sapwood, so sapwood contains, so sapwood, if you see the sapwood contains the cells like xylem parenchyma will be present, and you also have the xylem vessels. So xylem parenchyma will be present and xylem vessels. So in sapwood what happens is xylem parenchyma cells will be present and xylem vessels both will be present. But in case of the hardwood, if you see the hardwood, the majority of them are xylem vessels. Majority of them are xylem vessels. So these xylem vessels, these become dead. So these become dead actually. No doubt these are dead cells and they become further dead. So what happens is here, so conduction. So conduction in case of the trees, in case of the dichotomy, conduction of food material takes place with help of the xylem parenchyma in such case. So functional xylem. So which is the functional xylem means here the wood that is having the xylem parenchyma, that we have to take it as a functional xylem. So based on that your answer is, so sapwood will be our answer actually. So in a dichotomy what happens is secondary growth takes place. Whenever secondary growth takes place, the deposition of the wood will be taking place. The wood is of two types, that is sapwood and as well as the hardwood. So the hardwood is rich in the xylem elements such as xylem vessels, and they are dead and they won't conduct any food materials in case of the hardwood. So sapwood contains, no doubt sapwood also contains some amount of xylem parenchyma and xylem vessels, and that sapwood conducts the water actually. Okay, sapwood actually conducts the water, whereas hardwood does not conduct any water. So because of that reason, which is the functional xylem means we have to take answer as a sapwood is the answer as a functional xylem. Okay. So that is about the question. Let's move to the next question. So identify. So question is asking to identify the parts. So identify the parts labeled as 1, 2, 3, 4. So question is asking to identify the parts that is labeled as 1, 2, 3, 4. So let us see the parts of this particular diagram that is 1, 2, 3, 4. Let us see the parts. So this particular diagram, if you see this particular diagram, this is a, so what is this diagram? So this is nothing but, this is showing, this diagram is a bacteriophase. It is a bacteriophase. So this is a bacteriophase actually. So bacteriophase, it is called as a bacteriophase. It is a viral particle. It is a virus, so which infects, virus that infect or that attack. So that attack bacteria. So virus that infect or attack bacteria. Such type of virus particles are called as a bacteriophase. So if you see the shape of this bacteriophase, the shape of the bacteriophase is called as a, it is a tadpole shape. If you see the shape, it is a tadpole shape. So tadpole shaped virus is the bacteriophase. Okay. So if you see this, so this part is called as, so this part is a head part. So this part is called as a head part. It is made up of, it is surrounded by a protein structure. It is made up of protein that is called as a capsid. So that is the head part. Then no doubt it is a head part. Then if you see the this one, so fourth here, so this one fourth, this will be a collar. Okay, this part here it will be a collar part. It will be a collar. Then if you see the one, so this one will be a, this is a sheet. Then this will be a tail fiber. This will be a tail fiber. So from the given question, if you see the given organism is, it is a bacteriophase. What is a bacteriophase? It is a virus particle which infects only bacteria. The virus particle which infects only bacteria is called as a bacteriophase. So bacteriophase actually has three different main structures: head part, neck part and tail part. Okay, with help of tail fibers. In tail part, tail fibers are present. With the tail fibers, it attacks bacteria and sits on the bacteria. So, and genetic material is present inside the head part and inside the capsule. So based on that, if you see our answer will be, this is our answer actually. So no doubt about it, this will be our answer. So let's move to the next question. So the chondriosome. So next question is asking about the chondriosome. So chondriosome was discovered by which scientist? That is the question. So chondriosome is also called as, it is also called as mitochondria. Chondriosome is also called as mitochondria. So mitochondria was first discovered by Benda. So Benda is the person. So that is the reason we have to take answer as a here. This is the Benda. So that is our answer. Let us see the next question. Select the correct steps. Select the correct steps involved in the amplification of the gene. So this amplification of gene steps were given here. That method is called as a PCR method. What is the full form of PCR? Means it is the full form is polymerization actually, or polymerized chain reaction. So polymerized or polymerization chain reaction, PCR. Okay. So in that PCR there are different steps are present. So first step, so for example this is the double strand DNA in which two strands are present. The first step is denaturation. First denaturation takes place. Whenever denaturation takes place, the two strands will be getting separated. Two strands separates in this manner. So that process is called as a first step is called as a denaturation. Then after that we will add primers. We will add primers here. So that process is called as a annealing. Next step is called as a annealing. After addition of primers, then what happens is, with help of the primers, the extension will be taking place. With help of the primers, now this box like structure is a primer. With help of the primer, extension will be taking place. Now third step is extension. So finally there is several daughter DNA will be produced, but in this case we will be able to see two daughter DNA will be produced. So this is one DNA plus another DNA. So two daughter DNA is produced. This process is called as a amplification. So again this all together is called as one cycle. So this all steps together is called as a one cycle. So that again, so this two daughter DNA produced here will again enter into the second cycle. So like that this whole together is called as a polymerized chain reaction. This is called as a PCR, polymerized chain reaction occurs. So what are the major steps here is, first step is denaturation. So what is the denaturation? The two DNA strands which is initially bonded to each other, they get separated. So that is called denaturation. The separation takes place by heating of the DNA. So heating of DNA takes place between 80 to 90 degrees centigrade or 60 to 70 or something. Some books they write 70 to 80, some books they write 80 to 90 like that. So anyhow, so whatever may be, so denaturation of DNA will release the two DNA strands, and the two DNA strands, that process is called denaturation. After that, to the DNA strand the primer is attached. So this is the primer. So here also another primer. So primer is added. So primer helps in synthesis of the DNA. So that process is called extension. So anyway, the four major steps are denaturation, second one is called annealing, third one is called extension, fourth one is called as amplification. So based on that, so this will be our answer. So that will be our answer. So let us move to the next question. So question is asking about jet scheme. So jet scheme is connected with. Question is asking about the jet scheme. So jet scheme is connected with. So jet scheme is connected with the light reaction. So actually jet scheme is connected with the light reaction in a photosynthesis. It is connected with the light reaction. So where light reaction is taking place? Light reaction in photosynthesis. Jet scheme is connected with the light reaction in photosynthesis. It is connected with the light reaction in photosynthesis. So in photosynthesis, so two types of light reactions are taking place, that is cyclic photophosphorylation and non-cyclic photophosphorylation. So in non-cyclic photophosphorylation, if you see non-cyclic photophosphorylation, so light reaction involves two major process, that is cyclic photophosphorylation and non-cyclic photophosphorylation. So in non-cyclic photophosphorylation, what happens is, so two photosystems, so photosystem 2 and photosystem 1. So photosystem 1 and photosystem 2 are involved. So this photosystem 2 and photosystem 1 together undergo together undergo the or complete the light reaction. Such process is called as a jet stream. So they appear as a jet stream. So jet stream is shown by non-cyclic photophosphorylation. So that is the important part. Whereas in cyclic photophosphorylation, what happens in cyclic photophosphorylation, you have only one. So only one photosystem is involved. So that is the reason it is called cyclic photophosphorylation. Only one photosystem is involved. So that is why it is called as, and that will undergo the light reaction. So that is why it is called as a cyclic photophosphorylation. In non-cyclic photophosphorylation, what happens is two photosystems are involved and it appears as a jet stream. So anyhow, finally based on that our answer is non-cyclic photophosphorylation will be our answer. Next, let us see to the next question. Let us see the next question. Which of the following is the final step for genetically modified organisms? So which of the following is the final step for genetically modified organisms? So first step, not the final. So question is asking about the first step. So what is the first step in recombinant DNA technology? First step. So first step is to identify the gene of our interest. So let us see where is that point. So here, so identifying the DNA with the desirable gene. So very first step in recombinant DNA technology is you have to identify a gene, so which you want to amplify. So identify the gene which is essential for human day-to-day life. Okay, so identifying the desirable gene is the first step. So that is will be our answer. So identification of DNA with the desirable gene will be our answer. Next question. Yeast. So if you see the importance of yeast is no doubt it is unicellular. Yeast is a unicellular fungi. So all fungi are multicellular in nature, but what is the importance of yeast is a unicellular fungi. That is the importance of the yeast here actually. So yeast divides. So yeast completes cell cycle. So yeast also completes cell cycle. So yeast completes cell cycle in 60 minutes. So that is its importance. So yeast completes its cell cycle in 60 minutes. In 60 minutes it completes its cell cycle. For example, you see the bacteria like E. coli. If you see bacteria like E. coli, that is Escherichia coli, it completes cell cycle in 20 minutes. So here 24 hours. So more than 24 hours it is in case of the humans. In case of the humans, the cell cycle completes so in more than 24 hours. So more than 24 hours. So it is in case of the humans. So in case of the yeast, it is 60 minutes in case of the yeast, whereas E. coli bacteria completes its life cycle or completes its cell cycle. So completes its cell cycle in 20 minutes. So finally our answer is 60 minutes will be our answer. Next. Micropyle of seed is involved in. So micropyle. So next question let us see. So micropyle. So micropyle of seed is involved in passage of. So that is the question. So micropyle. Micropyle of seed is involved in the passage of. So micropyle. So if you see the micropyle. So micropyle is involved. So micropyle main function. So there is a micropyle. What is a micropyle? It is an opening. It is an opening of the ovule. So opening of the ovule. Okay, and after fertilization ovule is modified into seed. So opening of ovule. So that opening of ovule is called as a micropyle. So opening of some space. The opening of ovule is called as a micropyle. Then what happens is, so that the space also will be present in the seed. So that helps in absorption of water. So the main function of the micropyle is absorption of the water for germination of the seed. So when seed is about to germinate, it has to absorb water for germination of the seed. So that is the main function of the micropyle. So based on that we have to take the answer. So our answer will be water. So water will be our answer. Next. Colitotrichum. So colitotrichum. So no doubt colitotrichum is a fungus. So colitotrichum causes a disease that is called red rot of sugarcane. This is the disease that is caused by colitotrichum. So colitotrichum causes red rot of sugar actually. So red rot of sugar. So if you cut the sugarcane, then inside the sugarcane you will see the red color. So red color inside the sugarcane. So that is a red rot of sugarcane. So red rot of sugarcane is caused by the fungus. Name of the fungus is called as a Coltotrichum. So Coltotrichum is the name of the fungus which causes red rot of sugarcane actually. So let us see the next one, that is match the following. So let us see the match the following question. So pyruvic acid. So this is in the respiration. So this is about the respiration in plants. So in respiration in plants we have 3 steps. So first one is glycolysis, Krebs cycle, then we have electron transport chain or ETS, electron transport system. So these are 3 major steps in the aerobic respiration. So pyruvic acid. So pyruvic acid is no doubt it is an end product of glycolysis. Then acetyl-CoA. So acetyl-CoA is a connective link between the glycolysis and Krebs cycle. And ethyl alcohol is produced during anaerobic respiration. So ethyl alcohol is the end product of the anaerobic respiration. So that is how. Then carbon dioxide plus hydrols, H2O plus ATP is the end product of the respiration. So based on that we have to identify our answer. So pyruvic acid is option 4. So we can see here also option 4 is present. Then acetyl-CoA is the connective link between the glycolysis and Krebs cycle. Then ethyl alcohol is end product of aerobic respiration. So this will be our answer. Next question, let us see. Alkaloids. So alkaloids. So which are alkaloids here? If you see this, which of the alkaloid is? So morphine. So no doubt morphine is an alkaloid. So next. So no doubt is morphine is an alkaloid. Next, toxin. So resin is a toxin. Drugs. So drugs is vinblastine is a drug. Polymeric substance. Rubber is a polymeric substance. So based on that we have to identify the answer. So let us see this question. 1 is 2, then 2 is 3. No doubt it is correct. 3 is 1, vinblastine. 4 is 4. So this will be our answer. Next, which of the following is not differentiated? See, this is cambium. So cambium, this is all cambium. This is also having cambium. So this is also having cambium. So this is a cambium. So if you see the cambium, so what is the importance of cambium? So cambium contains, no doubt, they can become meristematic. Cambium contains meristematic cells. So whenever meristematic cells are present, they can undergo dedifferentiation. So no doubt, intracellular cambium can undergo dedifferentiation. Core cambium contain dedifferentiation. Intrafascicular cambium also can undergo dedifferentiation. So why they are able to undergo dedifferentiation is because of the presence of meristematic cells. Presence of meristematic cells make them undergo dedifferentiation. So there is only one dead cell here in these 4 options, that is tracheids. So tracheids are no doubt they are the dead cells. So they are dead cells. They are filled with a substance that is called as a lignin. So they are filled with a substance that is called as a lignin. So this is a dead tissue. That is the reason it cannot undergo dedifferentiation. So that is how we have to remember. Then, which of the following is not a bacterial disease? So citrus canker. So citrus canker is caused by bacteria that is called as a Xanthomonas citri. Actually, it is caused by bacteria called Xanthomonas citri. So blight of rice. So blight of rice is also caused by bacteria that is named as Xanthomonas coryzae. Then crown gala papil. Crown gala papil is caused by, that is also caused by bacteria disease, that is Agrobacterium tumefaciens. It is caused by Agrobacterium tumefaciens. So citrus canker is a bacterial disease. Blight of rice is also a bacterial disease. Crown gala papil is also blight of disease. The last one. So this is not a bacterial disease. This is a disease caused by nematodes. Nematode. So the name of the nematode is myeloid. So it is myeloid ignis. So it is a myeloid ignis, the name of the nematode. So finally, the 4th option, that is root knot in tobacco. Root knot in tobacco is a nematode disease. So this is a nematode disease, students. So that is end of today's class. So before ending the class, I would like to introduce about our Unacademy Plus subscription. So what I suggest you students, so I suggest you to take the Unacademy Plus class subscription students. So 1 month plan is about 2,250 rupees. So whereas you can also purchase the long term plans. I suggest you to purchase long term plan students so that you will be having a complete preparation. So you will be having complete preparation. So if you take the long term plan, so you can see here there is a long term plan. So the long term plan is of 6 months plan is available, that is 10,125 rupees, and 12 months plan is available, that is 12,375 rupees. I suggest you to take the long term plan students. And apart from that, so in plus classes, I am taking plus classes, and a person who take the subscription, they can join the plus classes. In plus classes, I am taking the topics of human physiology. So if you are interested to listen human physiology, you take the plus subscription. So 1 month plan of 2,250 rupees and 6 months plan of 10,125 rupees and 12 months plan of 12,375 rupees. If you subscribe this, you can join my classes of human physiology. So that is about our Unacademy Plus subscription students. So thank you all of you. So see you in the next class. So bye bye students all of you.